Okay, so yeah, I'll be speaking about GeoTA tool, the tool that is used to analyze spatially resolved transcriptomics, the method that was named the method of the year by Nature. Well, it was a great breakthrough when single cell was developed compared to bulk DNA. It allowed us to specifically study separate cells. But when we analyze data using single cell, we end up with something like this, just cells in an artificial dimensions. In this case, it's UMAP, but we never really know where exactly in our tissue these cells are located. And with spatial transcriptomics, finally, we have this opportunity to map the cells to our tissue. Like here, this example, this is the kidney, and this is normal kidney, injured kidney, and another injured kidney. And we can see that, for example, our cortex is comprised of a mixture of different cells that are located here, the glomerular or the collecting ducts. And our other cells, for example, proximal tubule cells, this third segment is located here. It's pretty much all the outer medulla is comprised only of this type of cell. But other than organs, we also can look at, for example, biofilm populations. This is the clustering of different functions that cells, individual cells in the colony do. And using spatial transcriptomics, it was possible to map them to the specific positions in the colony, which allowed to identify some hotspots for functions. For example, here we can see that this hotspot does the denitrification. Here we can see it's hotspot for high oxidative stress and so on. Of course, it's not the only application of spatial transcriptomics. It's used a lot in medicine, for example, in studies of cancer, because tumor is very heterogeneous and there are different clones. Some parts are evolving faster, and of course, we can see some differences in expression. Also, it's very useful to identify the invasive margin of the tumor and actually how the cells in this cancer interact with normal cells. And it was possible to build a bunch of expression atlases for healthy organs or the ones in disease. Also in molecular biology, it allowed to localize the individual molecules and, of course, study their expression inside the cell, for example, in cytosol or in nucleus. It also helped to understand better the chromatin organization and cell development. So there are different experimental platforms to study spatial single cell. We can separate them into microscopy-based methods like SECFISH, OSMFISH, or MERFISH, all the hybridization-based methods, and sequencing-based methods, for example, division. The difference is that for microscopy-based methods, since it's based on hybridization, we have the single cell or even single molecule resolution. And thus we can identify their locations if it's in cytosol or in nucleus or somewhere else. But we're restricted to only known genes, and we cannot study any novel ones. And we also can't analyze too many genes at the same time because we're restricted in number of colors, or they can just basically overlap and we won't see exactly where this molecule is. Sequencing-based methods are easier and cheaper, and they don't really need a lot of pictures made on microscope to study the locations. And we also can study novel genes that we don't know the sequence of, but it's not always a single cell resolution. And we will be focusing on vision, that is exactly not single cell resolution, but basically how vision works. We have a plate like this, and the capture area that is comprised of small spots. And each spot already has the barcodes, the spatial barcodes that will tell us where exactly this position is located. So we place our tissue above the capture area, and we treat it so the mRNA will ligate to our barcodes. And then we just proceed to sequence, and we get a sequence like this. So after sequencing, we end up with our matrix where we have gene counts per cell and cell coordinates. And of course, we can get some pictures to work with as well. So with the data I just showed, we can start our pipeline, and spatial transcriptomics is actually very similar at the beginning. It's very similar to single cell analysis. It also has the pre-processing steps, the dimension reduction, different clustering algorithms, and different algorithms for cluster-specific marker gene identification, cluster annotation. But Giotto also allows us to check, to play with clusters, to separate them or to merge them based on their similarity, and run enrichment. And this module, the spatial analysis, is what is really different and interesting for us. It starts with building spatial network, and then we can find spatially variable genes that have some spatial pattern and study co-expression and cell neighborhood. So I will focus only on this part. So we already did all the pre-processing and single cell analysis and end up with our artificial dimensions. In this case, it's UMAP. And we color them with latent clustering. Oh, in this case, we have 11 clusters. This is kidney sample. And what is different from single cell at this moment is that we already can map our cells on our tissue. And we see here that, for example, clusters 6 and 7 comprise the inner medulla, and classes 2 and 9 comprise the outer medulla, and the rest of the classes comprise the cortex, and it's really a mixture of cells. What we can see here is, for example, we can plot how many genes are expressed by which part of our tissue, and we see that medulla is expressing a lot more transcript than the cortex. So the first step in spatial analysis is to plot a spatial grid or network that will allow us to calculate the relationships between these parts. In case of grid, this is the expression will be summarized inside the grid. And in case of network, it will remain as a single cell or single spot resolution. And this allows us to run spatial gene search. And I wanted to show you an example of two genes, NAPSA and GPX3, which are both marker genes of proximal tubules. But you can see on these plots that they actually have completely different spatial expression patterns, right? We can see GPX3 expressed in the cortex and not expressed in the medulla, and NAPSA is expressed in outer medulla. So comparing it to already known cell types in the kidney, we can see that actually GPX3 is a marker gene for proximal tubules, but segments 1 and 2, which are located in the cortex. And NAPSA is more correlating with proximal tubules, the segment 3, which is located here in the outer medulla. So spatial analysis can help us understand marker genes better. Also, the next thing is spatial co-expression. So this actually allows us to identify the metagenes, a group of genes that are sharing same patterns of expression. And for example, for this data set, 8 metagenes were identified, and here we can see the correlation between these 8 metagenes. And here are the expression for the metagenes on our tissue. And you can see that they all have different patterns of expression. Some are specific to inner medulla, some are specific to outer medulla, some are specific, well, not quite, because here's a mixture of cells really, but they're specific to the cortex. And we can go from the metagene level to single gene level if we want to. And one more, maybe the most interesting thing, is how the cells interact. And in this case, it shows how really, well, the first step is to find the interaction changed genes. So the genes that actually change when their expression changes when there's interaction with different cells. And based on these genes, we can find cell types that are interacting within this cluster or with other clusters. And we can see, for example, that clusters 4, 5, 8, 1, and 3, they all are here. They aren't really correlating even to each other very well. And for example, cluster 10 and 11, these are small ones. They're located here and here. They have very strong interaction inside the cluster and not so much with other clusters. Well, they interact with each other as well. So a spatial single cell really gives us a lot of opportunities to study pretty much anywhere we can apply single cell. We can try applying spatial single cell, and it will give us more information at where exactly this function is happening, and a lot of information about expression patterns, spatial expression patterns. So that's it on the